organizers for inviting us. And I'm sure that I can also thank the organizers on behalf of the families, because we heard a huge cohorts here, but now I'm going to talk about a disease which is, at this point, there's less than 100 reported cases in the literature. So I think they would be very happy that we talk about their disease if they learn about that. So, and this is what I just literature review that a postdoc in the lab did, and this is, the disease, about 83 patients that he could actually find, or families with this reported ALG13-CDG. And then you could see that I just highlighted the ones, the most dominant phenotype is always like, neurological involvement and also seizures. But what is this ALG13-CDG? This is a gene, which encodes an enzyme, which is at the very beginning of building the glycan structures on proteins in the N-glycosylation process. And you can see that this is the enzyme, and it puts this glucanac molecule, the second one, on this glycan structure. And of course, if the enzyme is defective, then you will not be able to build that further, and then it actually results in some severe phenotypes. The most important unmet needs for this CDG is that you have no curative treatments. We don't really understand the disease at this point and the disease mechanisms and the pathobiology, and we don't know how this disease really causes the neurological symptoms. And then, of course, one of the questions in the literature was, because they could not really find glycosylation defects in this specific CDG when they looked at the traditional tissues that you can access, like fibroblasts or blood. So even the question was raised if ALG13-CDG is indeed a CDG with regard to its glycosylation phenotype. And I think the most important factor in having these questions and not yet solved is really the lack of model systems which have a translational value. They try to make animal models, and there is a mouse model, but they do not per se completely repeat the phenotype of the human disease, so they have their limitations. And this is where we actually turn to a system, a modeling system. And of course, you try to model a complex organ like the brain in an in vitro system. Of course, it has its own challenges, but as also previously, the talk Kilian mentioned, that sometimes, looking at the specific tissue can give you the answers. Because we thought that, as the presentation of the disease is so prominent in the central nervous system, we may only see glycosylation defects and problems in the central nervous system. And at this point, we have no access to the human brain, so there's no way to really directly study that, besides some imaging and other, maybe approaches, but not really the tissue. So we went to a new model system, which is the human forebrain organoids. They are about 2 millimeter big little spheroids, but they are actually representing a very early stage of the human development. But of course, there is limitations, because whatever we say here, it may not be completely or fully actually translatable to the complex human brain. So this is how an organoid looks like. Actually, they recapitulate the architectural features of the forebrain development. So I think we can use many aspects, of this organoid system to understand the disease. So actually, we hypothesized, as I said, that because of the presentation of ALG13-CDG, predominantly being neurological, we actually hypothesized that most of the glycosylation defects and also downstream problems will actually be only present in the neurons and also in these organoid systems. So we did a comprehensive kind of approach, So we developed these organoid models, and we did different omics studies. And the first one was the glycoproteomics, because we wanted to really show that these organoids, or this human brain kind of model or disease-relevant model, indeed shows a glycosylation defect. That was the first question to really solve. And here, some colleagues, Lindsay actually helped us, and they did a glycan imaging, and you can see on the BCDs, on the, for you, the right side, the control organoids, and then on the left side, you can see the ALG13-CDG organoid sections, and you could see that there is a difference, in the glycosylation pattern, and certain glycan species, which you can see here, were also significantly different between the control and ALG13-CDGs. And some of the glycan species like this this one here, we call them bifurcated glycans. These are very specific for the brain and the human brain, and this was also significantly affected in the ALG13-CDG cortical organoids. So this was the first kind of evidence that we see a glycosylation defect. And then we went further, and then also a postdoc at the Mayo Clinic, Rohit, he helped us, to do a glycoproteomic analysis. We did also the proteomic part. I'm not going to show the data of that. This is only the glycosylation part, or the glycoproteome part, when we looked at, the glycan structures on the proteins. And you can also see that we used to call it like a left shift, in the glycosylation in this volcano plot, and then you could see that there is indeed a significant disturbance, or actually downregulation of the glycosylation overall in these cortical organoids. So these studies actually already showed that indeed ALG13-CDG is a glycosylation disorder, but it may be very cell-specific or organ-specific, and maybe only affects the central nervous system. Again, sort of, connecting the clinical phenotype with the in vitro data. Then we also looked at some of the different, glycan structures, and then you could see that very clearly that the controls and then the ALG13, they have a really different kind of, abundance of the various glycan structures, like the high monos glycan structures, which are very, very specific for the brain. The brain usually actually has most of the glycan species belong to these high monos glycan species, and you can see that in the ALG13, this is now on the this part of the graphs. It's really downregulated, as well as the complex hybrid glycan structures. And it's no wonder, because this whole process is sort of, halted at the very beginning of the process, so you cannot really build the right glycan structures. And when we looked at, and that was also what we tried to further go into this, and then we took some of the gene panels, like the epilepsy gene panel. We also looked at some of the synapse-related glycopeptides and also extracellular matrix-related glycopeptides. And you can see that also in all these specific gene panels or pathway-related genes, we see a significant downregulation or sort of under-glycosylation, and especially in the epilepsy-related genes, which is the epilepsy panel, we can also see a significant defect or sort of impairment of glycosylation, and that could also explain the prominent phenotype, which is usually seizures in ALG13 individuals with ALG13-CDG. And several of the genes that we identified and also were peptides and also underglycosylated were already or have already been implicated in seizures. And then, so this one was the first part of the study, and now we could really show and then could answer the question, or that debate in the literature, if ALG13-CDG is indeed a congenital disorder of glycosylation and causes glycosylation defect, and also the hypoglycosylation of proteins, which we found they are also very important for brain development and function, but as I said, for seizure activity and other neurological presentations that we see in the ALG13 individuals. Then we went to a next step, and then we also tried to look into single-cell RNA sequencing, because in this case, from the brain organoids, we can really dissect, the cell-specific changes in gene expression in ALG13-CDG, and then Ramin again took lead on these studies, and then we have this is just the cluster identification or cluster sort of representation of the cell types that we could identify. These are pretty early stages. I mean, the organoids were about 60, 70, maybe 90 days. So that's very early in the development. But you could already see that we have different clusters, like GLOBergic. Already clusters are clearly present, glutamatergic, and you have also a lot of progenitors in this, the PC. These are also the progenitor cell types, and already the radioglial cells, which are usually very early in development and basically guide the migration of other neurons within the central nervous system. And when we looked at these different clusters, again, comparing the ALG13 and the controls, it is very clear that in all of these clusters, GLOBergic, and this is just a few examples, and glutamatergic, and also the radioglia, we actually see common themes, if you will: neuronal migration, genes associated with neuronal migration, neural excitability. They were all coming, in these different clusters. There were a few specific ones, like the GABAergic neurons. They also showed a very profound difference in the oxidative stress, which was not present in the other neuron cell types. So it may not be just, a organ-specific presentation or organ-specific pathobiology, but we may have to go into the cell-specific biology to understand fully the disease. And the next one, I think this is also an interesting approach, which also Ramin did, and this is a developmental trajectory. This is a pseudotime representation of the development of the different clusters, and you can see on the, like for you is the right side, the control pseudotime, and on the left side is the ALG13-CDG cortical organoid pseudotime. And I think it's just very easy to see when you look at, this graph. For example, if you look at the glutamatergic cell types, it's very different, This is actually delayed, or actually this is the normal development, but if you can see in the ALG13, this is somewhat, accelerated. So we have these differences. GABAergic neurons also show differences in the pseudotime. And then we also put that in a graph, and this box plot also shows these pseudotime differences, how these neurons develop compared between the ALG13-CDG cortical organoids and controls. And again, I think you can see that the GABAergic cell population is somewhat delayed in development compared to the controls. And if you look at the glutamatergic, it's just the opposite. They are somewhat accelerated in development. And we know at the end, in the, from human brain literature, but also from animal studies, that the development and the balance at the end, in the brain of glutamatergic and GABAergic neuron population is very critical for many functions, also for seizure activity, but also cognitive development and also cognitive functioning. And if you have a developmental kind of a difference, delayed GABAergic and maybe accelerated glutamatergic development in ALG13-CDG in the brain, that could actually cause later in life some differences or actually dysfunction in this GABA-glutamate balance. And these were maybe the most prominent changes. Some of the astrocyte progenitor cells are also were differentially sort of developing, so that could also be very important when we look at the pathobiology of ALG13-CDG. And then we did also some metabolomics, and this was somewhat surprising because we did some organoid work, in other CDGs, and we always saw, and also like in fibroblasts, a very robust change in metabolism, a very robust metabolic reprogramming. Interestingly enough, in these ALG13-CDG cortical organoids, we actually saw a very limited metabolic rewiring. Seems to be, less important, but there were a few metabolites like this HAXNAC, and we suppose that this is glucnac, but the method cannot really distinguish here glucose specifically, but also these two metabolites which were significantly downregulated in the ALG13 cortical organoids. And the accumulation of this glucnac, or it's only HAXNAC because the method cannot differentiate between the two, is of course very easy to understand, because if the enzyme cannot actually put a new, or actually the second glucnac on the glycan chain, then that precursor actually accumulates in the brain, and this is why we see that. The TMP and the septohallulose, they are also very important metabolites, one in the phosphate, pentose phosphate pathway, and the TMP is in the nucleotide synthesis, and we could see both downregulated. We still don't understand fully, how the ALG13-CDG, which is causing some glycan synthetic problems, and also this glucnac accumulation, as I said, how that really causes some differences or problems with the nucleotide synthesis. That is still something that we would like to understand, and how that contributes to the disease phenotype or the neurological phenotype that we see in ALG13. So the conclusion is that, as I said, that ALG13-CDG is a glycosylation defect which is specifically affecting the brain, and maybe the brain is the only tissue where you see significant differences in glycosylation or impaired glycosylation. I think we have a model for ALG13-CDG. with its limitations, but I think it's a new and interesting model to study further pathobiology. And also the pathways that we discovered, could also understand the pathobiology. And then in the lab, Irena is also a postdoc. And then when we moved to Mayo Clinic, then we got in contact some colleagues, and they have this very I mean, they use this very sophisticated, for me, sophisticated, maybe for many not, this ensemble of multiple drug repositioning approaches, so where you can actually put all your multi-omic data, and then you can identify potential targets. And then when we did that, and we put in, the proteomics data into this EMUDRA, we could actually identify certain targets, or pathways, which could be targeted by drugs, and then that could also help, improve the phenotype. And this is important because, as I said, in these individuals, we have no treatment whatsoever. And of course, in the lab, the next step would be to look into these specific pathways and then try to reverse the phenotype in these brain organoids, and thereby, providing some novel therapies for this really ultra-rare disease and underserved disease. And that was the presentation, and of course a lot of people contributed, Mayo Clinic, and some actually, because we just moved to Mount Sinai in New York. Many of these people, like Ramin and then also Graham, came with us. And Emory University, I have to mention Stephen's name. His group was very helpful and instrumental for us, to establish the organoid models. And the Medical University in South Carolina, who did the glycan imaging on the brain organoids. And we have the funding sources who actually helped us, and really we thank, the Rare Disease Clinical Research Network, because they provide us, a big umbrella organization, and within that, we have the Frontiers of Congenital Disorders of Glycosylation, where we have multiple CDGs at this point. More than 100 different CDGs included in this study, also the ALG13-CDGs. And thank you for listening.
So in the interest of time, as we went way past the 15 minutes mark, before we finish this session, I want to remind you that right now there is a 30-minute break, and then there will be 9 concurrent sessions. After this 9 concurrent session at 8:15, you are all invited to join us for an opening mixer, and so with some drinks and some finger food. So, but please join me to thank Vanessa, Conrad, Andrew, Kilian, Catherine, and Thomas. Thank you.
Thank you.